Good morning, Fernando. Myself, Dr. Ronnie S. Emmanuel. I will be dealing the veterinary physiology portions for your understanding. I have given a very brief profile about me and I have added my email address so that you can contact me if at all any doubt arises in the upcoming sections. Without any delay, let's go on to the sections today. Section today, we are going to deal the blood, the fluid connective tissue of the body. Do you people know who this person is? I have added he is the man with the golden arm. We will see about him at the last slide. Now you can make some assumptions or you may be having the correct answer with you. So throughout this lecture series, we will deal what are the general features, uh, the leukocytes, something about the erythrocytes or the RBCs, the hemostasis, which is otherwise the prevention of the loss of blood and plasma. These main headings will be dealt in our upcoming slides. The general characteristics of the blood. If we see, the blood is having a fluid portion which is otherwise called the plasma, and it does have cellular elements. The cellular elements comprised of the RBCs or the erythrocytes, the WBCs or the white blood cells or the leukocytes, and the platelets. The plasma is around 55 to 70 percentage of the blood volume, and the cellular elements makes up 30 to 45 percentage of the blood volume. The first characteristic is the specific gravity of the blood. If you see the specific gravity of the blood, the horse is having a 1.060. It is more denser than the water. If you see the pig, it is also 1.060. The least dense one is the sheep blood, which is 1.042. The color of the blood. In every mammals, the color of the blood is red in color. That is also true with birds and reptiles. The blood is red due to the presence of hemoglobin. But the color of the plasma can vary between various species. The usual color of plasma is yellow to colorless. What makes the plasma yellowish? It is because of the presence of bilirubin. If the bilirubin is more in present, it will be more yellow. So bilirubin is actually a degradation product of hemoglobin, which we will see in the further slides. In case of cats, dogs, sheep, and goats, the color of plasma will be slightly yellow or colorless. In case of cow, it will be more darker or stronger yellow in color in comparison to the previous species. In case of horse, the color of the plasma is very deep yellow. Next characteristic is the blood volume. The blood makes up 8 to 10 percentage of the body weight. How to calculate the blood volume? Blood volume is equal to PV divided by 1 minus HCT, where PV stands for plasma volume, HCT stands for the hematocrit or the packed cell volume. Packed cell volume is nothing other than the volume of the cells in comparison to the total volume of the blood. The blood pH, it is another characteristic. It is otherwise called the reaction of the blood. We know pH is the uh, negative power of the logarithm of the hydrogen ion concentration. So the blood also has got a pH. The normal blood pH is 7.4, but the venous blood is slightly acidic. Why? Because the presence of carbon dioxide, which is an acidic type of gas. And here I have added the arterial blood pH of various species. We can see that all species are having a blood pH of 7.3 5 to 7.5, almost in and around the same range of 7.4. Next is the erythrocyte or the RBC, which is the next important subheading. We will discuss about it in detail. The numbers of RBC, I have highlighted the numbers of RBCs in case of goats as in and in chicken. The numbers are depicted as 10 raised to 6 cells, which means million cells per microliter. We can see that goat RBCs are the highest number. It is 13 into 10 raised to 6. 13 million RBCs are present per microliter of blood. While in case of chicken, it is only 3 into 10 raised to 6 cells per microliter. That means only 3 million cells are present per microliter of blood. And other species will be in between this 30 into 3, we can see. In case of the mammals, we can see that pig is having the least amount of RBCs. It is 6.5 into 10 raised to 6 cells per microliter. How will we estimate? Normally, we estimate the RBC number by a hemocytometer, which you probably might have seen in your practical classes, or there are some automated cell counting machines are there. If anemia is a condition in which there is a decrease in the RBC number than the usual amount, or the hemoglobin concentration is reduced, you might have heard that in schools and in nurseries, children will be given, or girl students will be given some iron tablets, or the students in general will be iron tablets because they might have anemia. And the polycythemia is a condition in which there is an elevation in the RBC numbers. So let's see what is anemia. This is a very brief description about what is anemia. Anemia is basically the reduction in the RBC numbers and or the hemoglobin concentration. Either RBC number will decrease or the hemoglobin concentration will decrease, or both of them together can decrease. This is the classification of the anemia. Anemia can happen due to a defective blood formation. There are two subtypes of it: aplastic anemia and anemic anemia. Aplastic anemia means that there is no or very little RBC production. We know that these RBCs are produced mostly in the bone marrow, right? So this can happen due to some nutritional factors or bone marrow damage, some radiation toxicity. Some people who have uh, so much uh, so much exposed to the radiation, they might have anemia because of this aplastic type of anemia where no RBCs are produced. Or next is the anemic anemia. Anemic anemia can happen due to iron deficiency. In that case, microcytic hypochromic anemia can happen, which means that microcytic means the cell size will be very small. Hypochromic, which means that the hemoglobin concentration will be very less, so that the color of this RBCs, when it is stained with the usual Leishman type of stains, we know that when we look through the slides, RBC will be like red in color. But in a hypochromic type of anemia, that reddish color will be lost. Then vitamin B12 deficiency, which produces macrocytic hyperchromic anemia, which is otherwise called the megaloblastic anemia. Why? Because it's very large in size, megalo, large size. Then next is a Casis intrinsic factor deficiency. Casis intrinsic factor deficiency causes pernicious anemia. This type of anemia is normocytic normochropic, which means that the size will be normal and the color will be also normal. So the problem is that there will be a lesser number of cells will be there. RBC's total number will be reduced. This is seen in a Casis intrinsic factor deficiency. We know that that is required for vitamin B12 absorption in case of animals. Next type of anemia is to blood loss or RBC destruction. If hemolytic anemia means that hemolysis occur, the destruction of the RBCs occur. So the total number of RBCs reduces. So the remaining RBCs will be normocytic normochromic, which looks apparently normal in both their size as well as their color. So one is hemolytic anemia and the next is hemorrhage, a type of excessive bleeding. If hemorrhage is occurring in large amounts, still the remaining type of RBCs will be looking normal. So that is normocytic normochromic type of anemia. Further, the polycythemia. Polycythemia have discussed that it is absolute, means it is the increase in case of the RBCs. It can be either absolute polycythemia or relative polycythemia. Absolute polycythemia means that increase in the red cell mass without a decrease in the plasma volume, which means that the plasma volume will be remaining constant, but the cells as such, cell number will increase. This is seen in case of a tumor causing increased erythropoietin. We will see what is the importance of erythropoietin in the upcoming slides. So the erythropoietin is basically a hormone that is produced in the kidneys. So erythropoietin is required for the production of RBCs. If erythropoietin is produced in large amounts, the RBC will be also produced in large amount. So this is called the absolute polycythemia. Next is the relative polycythemia. This is the increase in the red cell mass with a decrease in the plasma volume. Just imagine you are having a glass of water and you are having a little bit of adding little bit of salt to it. So if you boil it, the water will reduce, right? Meanwhile, the concentration of the salt will automatically increase. Why? Because the water is getting evaporated. The same way, in case of a dehydrated animal, the volume of the blood as such will decrease. The plasma volume will decrease, but the cell will not reduce in number, right? So this is relative polycythemia. And another one issue is the polycythemia vera, which is a pathological. Pathological means an abnormal increase in the RBC number due to a myeloproliferative disorder, as in myeloid leukemia. Leukemia is a type of blood cancer. So myeloproliferative disorders means that in the RBCs are produced inside the bone marrow. So if some issues causing a tumor like situation arises in the bone marrow, there will be an excessive production of the RBC. This is called the polycythemia vera. Next feature of RBC is the shape. You can see that sheep, dog, cattle are having typical biconcave shape. Horses and cats are having a shallow biconcave. Goats are having a spherical. Camel are having elliptical. Deer, sickle shape. I think the deer RBCs are also called the depranocytes. In case of birds and amphibians, they are elliptical and nucleated. This question might ask, if you have seen in clinics the shape of the RBCs of dog, they are very beautiful. They are typically biconcave. But if you get some uh, bird sample, you will see that they will be looking like very elliptical and they will be having nucleus. Normally, RBCs are devoid of nucleus, right? So, another feature of RBC is the plasticity. Plasticity is the abilities of RBC to change its shape when they passes through a small lumen. We know that RBCs are going through small blood vessels, and the lowest size of this blood vessel are seen in the case of capillaries. When RBCs are passing through the capillaries, which means that when it is passing through a small lumen, the shape of the RBC can change to a little bit so that it can easily pass through it. When the RBCs might have been wider, the RBCs when passing through a small lumen will become narrower. This is called the plasticity. Next feature is the poikilocytosis, which means that presence of abnormally shaped RBCs. Usually all RBCs are typical biconcave shape. If the RBCs are in a in a specific species, if the RBC if we collect and they are of different shapes, it is called poikilocytosis. This is the size of RBC. We can see that dog is a species with the largest size of RBC, while goat is a species which is having the smallest size of RBC. These are two data that I have collected from Duke's Physiology itself, but the tabular form is showing 4.1 and the text is showing 4.8. But still, if somebody asks you in an exam which species is having the smallest RBC, you can come up with goat and sheep. If specifically goat and sheep are given as two different options, I always suggest to go with goat because many textbooks says goat is the goat having the smallest size of RBCs, but both are given as true. And next feature is the anisocytosis. Anisocytosis is a condition characterized by the presence of RBCs of unequal sizes. The size of RBCs, uniso, means different sizes of RBCs. Further, what are RBCs composed of? We have seen what is blood composed of. Further, the RBCs are composed of 62 to 72 percentage of water and 35 percentage of RBC are solid. Out of this 35% is solid, 95 percentage of it is hemoglobin only, which is the pigment of RBC. Then the remaining solids are lipids, which are composed of lecithin, cephalin, sphingomyelin. All of this will be studied in the biochemistry. Free cholesterol, cholesterol esters, vitamins, various enzymes. Two important enzymes are carbonic anhydrase, cholinesterase, and some minerals. Potassium is the chief intracellular cation of most species, except cat, cow, dog, and sheep. Please remember it. Chloride ion is the principal intracellular anion. We will study in renal physiology what are the concentrations of uh, each of these cations and anions inside the cell. This will be dealt in the further sections as well, for upcoming classes as well, I'm saying. And the surface area of RBCs. It is relatively constant in case of all mammals. It is from 57 to 67 meters square per kilogram. And in case of birds, it is 44 meters square per kilogram. Next, three important things. Erythrocyte indices. This you might have seen in your practical sections as well. First one is the mean corpuscular volume. Second, the mean corpuscular hemoglobin. And three, the mean corpuscular hemoglobin concentration. These three are very important. First, the mean corpuscular volume. It is corpuscular means cell. Just imagine like that. It is basically the average volume of a red blood cell. How much a red blood cell will have the volume? The equation is hematocrit divided by RBC into 10. The result will come in femtoliters. Next is the mean corpuscular hemoglobin, which is the average amount of hemoglobin in a single red blood cell, which is hemoglobin divided by hemoglobin concentration divided by the number of RBCs into 10. So you will get the result in picogram. Please study the units, they are important. The mean corpuscular hemoglobin concentration, which is the average concentration of hemoglobin in a given volume of packed red blood cells. Here you have seen in the mean corpuscular hemoglobin, it is RBC that comes the denominator. But here the hematocrit or the PCV comes as the denominator. So this is important. If a concentration, mean corpuscular MCHC, mean corpuscular hemoglobin concentration, here the hematocrit will come as the denominator. And you will get the result in gram per deciliter or gram percentage. Here the RBC concentration will be in millions per microliter, hemoglobin concentration will be gram per deciliter, and hematocrit or the PCV is the packed cell volume, is the volume percentage of the red blood cells. I have given you three questions. You can stop or take a screenshot of this and do it. And I would definitely love to see your answers. Please send me in my mail if you would like to. I would definitely give it a reply. See, the RBCs has to be produced, right? So erythropoiesis is the process of the production of RBCs. It's very important. The prenatal means before the birth, the RBCs are produced in liver, spleen, and bone marrow, right? But after birth, the RBCs will be produced in the skeleton, which means inside your bone, and that too, the bone of the appendicular system. Appendicular system means your limb, two forelimb, two hind limb, right? And 65% of the production of the RBCs will be happening there. And in case of little bit, 35% of the RBC production will be happening in case of the axial system, which means the head, the thorax, and as such. The production of RBCs is a continuous process. If you stop the production of RBC, how will you live? Because RBC will get destroyed after some days. Na? Usually it is there for 120 days. After 120 days, they will get destroyed. In case of human being, I am saying. So RBCs has to be produced from the stem cells, which means stem cell is a cell which can replicate, which can divide and give rise to progenitor cells, means, and the stem cells will be itself. It is basically a progenitor type of cell. For example, there is a cell called a cell A. The cell A will multiply and it produces a cell A and cell B, and the cell B will multiply and the cell A will remain again as the stem cell, and from which again another cell A will produce, another cell B will produce. It is very easy. Uh, Colony forming units are the first uh, stem cells for this production of RBCs or the erythropoiesis. From them, proerythroblast will be produced, which will give rise to basophilic erythroblast, which will give rise to polychromatophilic erythroblast, leading to orthochromatophilic erythroblast and reticulocyte and the red blood cell. You have to study it. You have to write it and study it. Otherwise, you will definitely forget it because everything will get confused. There are 4, 5, 6 names are there, right? So please write it and study. And there is a rubriblast, another name of proerythroblast. For prorubricite is another name for a basophilic erythroblast. Rubricite is another name for polychromatophilic erythroblast. Metarubricite is another name of orthochromatophilic erythroblast. Reticulocyte and the RBCs or the red blood cells or the erythrocyte. See, proerythroblast, basophilic erythroblast, polychromatophilic erythroblast, orthochromatophilic erythroblast, reticulocyte, erythrocyte. Here you can see that when there is a low blood oxygen, what is the function of RBCs in fact? RBC function is to carry the oxygen, right? So if there is a reduced amount of oxygen, there will be production of the erythropoietin from both the liver as well as kidney. But kidney erythropoietin, you can see that the arrow is more stronger in case of kidney. So the kidney erythropoietin production is very high in comparison to the liver erythropoietin production, and they will reach the erythropoietin will reach the bloodstream, and there is an increased production of RBCs from the bone marrow, and then there will be an increased amount of oxygen carrying capacity, and this will cause a reduction in the case of reduction in the production of erythropoietin. This is a negative feedback. This is a positive. This is very easy and simple diagram. Please keep it in your mind. Reticulocytes are seen in the circulation which are having polyribosomes, brackets, I have written polysomes, and mRNA molecule. Basically, you know that in a adult RBC, you will not see any genetic material. Nucleus is absent, right? In case of reticulocyte, there will be a little bit of mRNA will be seen and ribosomes will be also seen. What is the function of mRNA and ribosome? It is nothing other than the production of protein. So protein production is still undergoing in reticulocyte. If at all the RBC lifespan is very short or more RBCs are produced, if a hemorrhage is there or if the sudden blood loss is there, the body has to produce more RBCs. At that time, reticulocytes more amount of reticulocytes will be seen in case of in such situations, because the body is producing more and more amount of new RBCs, so immature RBCs. Reticulocytes are basically immature RBCs. This immature RBCs will be seen in blood in more amount. I have discussed already about erythropoietin. The lifespan of the RBCs. Every cell that is produced will be destroyed in one time or the other. So the lifespan of RBCs is given in the tabular form. You can see horses is having 140 to 150 days, adult ruminants 125 to 160 days, pigs 75 to 95 days, dogs 100 to 120 days, almost similar to human beings, chicken 20 to 30 days. What happens once this RBC gets very old? This RBC membrane becomes more fragile and rigid. I have earlier said that RBCs usually have a plasticity. They will lose this plasticity and they will become this discocyte. You see, this is apparently a normal looking RBC. This will become very rigid, short RBCs. This is called discocyte, and the discocytes will finally become spherocytes and they will get destroyed. We will see in the upcoming slide how it is happening. The RBCs which are very old has to get destroyed. So the destruction of the RBCs is called the hemolysis. The hemolysis occurs in both inside the blood vessel, intravascular hemolysis, which accounts for the 10% of the RBC destruction, whereas the extravascular hemolysis, which occurs outside the blood vessel, that happens in case of 90% of the RBC destruction. In both type of hemolysis, the RBCs are phagocytized by the mononuclear phagosystem cells, which are seen in case of spleen, liver, and bone marrow. Here you can see what is happening. The RBCs are composed of, as I mentioned, hemoglobin as well as the water. So hemoglobin is composed of heme and the globin part, which we will see in the further slides. Within the mononuclear phagosystem cells, the hemoglobin is separated to the globin and the heme. The heme is again having iron component, which is also separated, and the remaining is the biliverdin. The biliverdin is a green pigment and the bilirubin is an yellow pigment. Up to bilirubin formation, it is occurring in the mononuclear phagosystem cell. The globin and the iron, both of them will not be wasted. Both of them will be reused again for the hemoglobin production. Here the iron that is separated from the heme can either be stored in the MPS cells as ferritin form or the hemosiderin form, or it can be transferred to plasma, where it will combine with a plasma protein called the apotransferrin. When it combines with the iron, it forms a transferrin. This is used for the new hemoglobin synthesis. And the remaining we can see that the free bilirubin, the bilirubin that is produced within the MPS cell, it is water insoluble. So it cannot be dissolved in the plasma, which is mostly water. So it has to bond with the albumin. It combines with the albumin and then it is transported to the liver. In the liver, this bilirubin conjugate or bilirubin uh, chemically combines with the glucuronic acid and it forms the bilirubin diglucuronide. The bilirubin diglucuronide, this is water soluble and this is secreted into the bile and thus it reaches the intestine. We know the bile is to the hepatic bile duct, biliary duct, it reaches the intestine and the large intestinal bacteria again converts this bilirubin diglucuronide to urobilinogen. Urobilinogen is formed within the large intestine. This oxidized form of urobilinogen and stercobilinogen. This urobilinogen is converted to stercobilinogen, which is also by the intestinal bacteria. They are the urobilin and stercobilin respectively. And this is the one that this urobilin and stercobilin are the ones that give the feces or the excreta its typical yellowish or brownish color. But a little bit of the urobilinogen is absorbed back. It is absorbed back into the liver. How it is absorbed back into the liver? By the portal circulation. You might have studied about it in the anatomy that it is absorbed back by the enterohepatic portal system, and it will reach the liver again, and from the liver it is again secreted back from the bile, uh, and the urobilinogen again reaches the large intestine. A part of this enterohepatic circulation which escapes the this excretion, it will go to the general circulation. It will go to the general circulation, and thus the part which is going to the general circulation will reach the kidney. In the kidney, some of the urobilinogen that bypasses this portal circulation will reach the general circulation, and it reaches the kidney, and where it is converted to, it is oxidized to form the urochrome or urobilin, which is giving the typical yellow color to the urine. There are some uh, abnormal condition, the icterus or jaundice. You might have seen or heard about this disease condition, which is basically a symptom. When there is an increased amount of bilirubin in plasma, it produces an yellowing of the mucous membrane. For example, when the bile duct becomes blocked, the bilirubin glucuronide or the soluble bilirubin will spill over to the plasma, and it will cause the yellowing of the mucous membrane. In case of intravascular hemolysis, the hemoglobin is bound to haptoglobin. Hemoglobinemia, it is an elevated hemoglobin due to more intravascular hemolysis and causing a reddish appearance of the plasma. Hemoglobinuria is the presence of hemoglobin in urine. Here I have added two photos of this condition. If the hemoglobin is present in urine, the urine will be more darker in color. Next, we have discussed about this hemoglobin a few times. So what is basically the hemoglobin? Hemoglobin is basically a protein, which is a conjugated protein, which is having which is a complex protein with heme group. It has a globin molecule. It is accounting for 1/3 of the erythrocyte content. The molecular weight, it is about 67,000 Dalton or 67 kDa. The proteins are expressed in Dalton sizes. It is having 4 heme groups. You can see, and it is having one molecule of globin. The globin is the protein component. Globin has 2 alpha chains and 2 beta chains. You see this spiral structures. You can see 4 different colors are there: one green, orange, blue color, another one with the violet or purple color. You can see this is the globin part, and within the globin part you can see there are some small like typical this porphyrin groups are there. This makes the heme group 1, 2, 3, 4. There are 4 heme groups are there, and each heme group will contain one iron atom. Therefore, one hemoglobin will contain 4 iron atoms. The iron valence is +2. Whether it is having an oxygen or it is not having an oxygen, it must have, it must be having the valence status +2. Please don't think that when it is getting oxygenated or when it is deoxygenated, the valence changes. No, the valence doesn't change. Because of the presence of this iron, the hemoglobin transport of the oxygen, it is more than 60 times if it were not present. The presence of hemoglobin increases, or the presence of iron in the hemoglobin increases the blood transport of oxygen by 60 times. You can see that heme, this heme is held to a specific polypeptide chain. One of this protein globin by a cysteine. A cysteine residue will be there, and a imidazole residue will be there. And imidazole residue will be there. Cysteine residue and imidazole residue. This will form a bridge. See, bridge formation. And this is the one that keeps this heme in heme. It is bound to the our this peptide chain or the protein by the cysteine residue and the imidazole residue of the peptide or the protein. We can see some forms of hemoglobin, where the hemoglobin and the carbon monoxide hemoglobin are the abnormal forms, where the myoglobin is the normal form. The methemoglobin is the oxidized form of hemoglobin, where the valence state of the ion will be 3 plus instead of the usual 2 plus. It cannot transport oxygen because it is 3 plus, and because of this the blood color will be a chocolate brown. It usually happens in case of nitrite poisoning. Next is the carbon monoxide hemoglobin or the carboxy hemoglobin. We know that the affinity of the carbon monoxide for hemoglobin is 200 times that of oxygen, because of which there is competition between carbon monoxide and oxygen for the hemoglobin. But the affinity is more in case of carbon monoxide, so the carbon monoxide will occupy the hemoglobin, and there will be no oxygen cannot bind to the hemoglobin. Because of this, there will be no oxygen availability to the tissue, and the blood color in such situation will be bright cherry red. And next is the myoglobin. The myoglobin is hemoglobin of the muscle. Instead of the 4 chains, we have seen that the globin of the globin part of the hemoglobin is having 4 chains, 2 alpha and 2 beta. Here only one polypeptide chain will be present, and because of this there will be only one heme group will be present. Here you can see the abnormal color of this is the usual blood, and this is a chocolate brown colored methemoglobin. Next important cells or the next important subheading is the white blood cell, the WBC or the leukocytes. The WBCs are of two broad categories. They can be either the granulocytes or they can be the agranulocytes. Granulocytes means the cells which are having granules in them, and agranulocytes are the cells which does not have granules in them. The granulocytes can be eosinophils, neutrophils, or basophils. And basophils. There are 3 types of granulocytes, and the agranulocytes can be lymphocytes, monocytes, and the lymphocytes have 2 broad categories, T cells and B cells. The myeloid stem cells and lymphoid stem cells are the stem cells for the formation of this white blood cells. From the myeloid stem cells, which are found in the bone marrow, there are 2 types, myoblast and monoblast. The myoblasts are required for the formation of the granulocyte, whereas the monoblasts are required for the formation of the monocyte, where in case of the lymphoid stem cells, the lymphoblasts are the stem cells. They are found in the lymph tissue. The lymph tissues are found in various lymph nodes, spleen, liver, and bone marrow, and various lymphoid clusters. You have probably heard about that in your anatomy classes. They are found, the lymphoid cells are found in these tissues. Now let's see some numerical data, where I have given you the list of the species with their ratio of WBC to RBC, that percentage of neutrophils, percentage of lymphocytes, and the total leukocytes in 10 raised to 3 cells per microliter. We already know that in case of RBCs, the numbers was in million cells per microliter, but here the cell numbers are in 1000 cells per microliter. 10 raised to 3 means 1000. We can see that in case of goats, the ratio of WBC to RBC is the most highest, 1 is to 300. Where in case of chicken, it is only 1 is to 100. Please study at least the highest and the least. And if you notice, in case of some species, the neutrophil counts or the neutrophil percentages will be more in comparison to the lymphocytes. For example, in case of goats, the lymphocytes are higher. Sheep, lymphocyte percentages are higher. Cattle, lymphocyte percentages are higher. Swine also, lymphocyte percentages are higher. And chicken also the same thing. But in case of dogs and cats, you can see that the percentage of the neutrophils will be always higher. Please remember that. Okay. This is same, almost same situation in case of human beings also. And if you see the total leukocyte counts also, you can see that in sheep and cattle it is lesser. But in case of chicken, the total leukocyte counts are always higher, 20 to 30% in comparison to all other species. There are other white blood cells as well. These are about neutrophils and lymphocytes. But monocyte, eosinophil, basophils are also seen in blood of these animals, but their percentages are far lesser in compared to the others. That means in comparison to neutrophils and lymphocytes, the percentage of monocyte and eosinophil and basophil is less only. Monocyte will be like 5 to 6%. In case of chicken, it will be higher, 10%. The eosinophil counts, it is 2 to 5%. In case of chicken, again, it is higher, 3 to 8%. The basophils, it is always less than 1% of in case of every mammals, but it will be in chicken, it will be 1 to 4%, which is higher in case of in comparison with mammals. If we see the leukocytosis, it is an increase in the leukocyte number. Leukopenia is a decrease in the leukocyte number. And leukemia, we have heard this word multiple times in our life, I suppose. It is a cancer of the WBCs and it is characterized by leukocytosis. Differential leukocyte count or the DLC, it is basically we are determining what is the percentage of each type of WBC, like how much percentage of eosinophil is there, how much percentage of neutrophils are there. You will count 200 or 100 leukocytes or WBCs, and out of this 200 or 100 cells, how many neutrophils are there, how many eosinophils are there, how many basophils are there, and with this comparison we will get the differential leukocyte count. If you see the neutrophils, the first type of or the one of the most important type of the WBCs, the nucleus of the mature neutrophils is divided into lobe, and usually there will be 5 lobe, and they will be stained blue or purple by the Leishman stain. They have abundant and fine granular cytoplasm which takes up the neutral stain. That's why if you see the neutrophil, neutro, which means that they will take the neutral stain, and because of this there will be no specific color will be seen. And there are granules within the cytoplasm. These granules store lysosomes which contain hydrolytic enzymes, proteolytic enzymes, and lipases to digest the invading organism or the pathogen. These are the two important type of the neutrophilic granules. First is the azurophilic granule or the primary granule. They are the lysosomes of the neutrophil. They contain myeloperoxidase, defensin, cathepsin, etc., which are required for the destruction of the invading organism. While the specific granule or the secondary granule, they contain hydrogen peroxide, which requires the peroxidase enzyme for its activation, the collagenase, and most importantly, it contains an iron binding protein called lactoferrin. Because of the presence of lactoferrin, the bacteria require iron for their action, for their survival. But if the neutrophil is going to attack it, the neutrophil has got lactoferrin in them, and the presence of lactoferrin makes the bacteria deprived of the iron, so the bacteria cannot survive. Next, let's see how these neutrophils can attack the invading organism or the pathogen by the five properties of the neutrophils. First property, they are having the chemotaxis or the chemically attracting property. Means that the neutrophils can release by themselves and they can also get attracted to some chemical substances, and they can easily respond to it. Second, when an organism is coming, they may be outside the blood vessel, so the neutrophils has to reach outside the blood vessel. So they are flowing through our blood vessel. So they have to get attached to the blood vessel so that they can come out of the blood vessel. So this property of getting attached or adhered to the blood vessel or the vascular endothelium, it is called the margination. And the third is the diapedesis, which means the neutrophils can squeeze through the endothelial openings. Between the blood vessels, some opening will be there. So the neutrophils can come out of through this opening. Fourth, the neutrophils have amoeboid movement. Like an amoeba, the neutrophils have the movement, which is absent in case of RBCs. And last, when the neutrophils arrive at the inflamed site, they will phagocytase. Phagocytase means that they can engulf the bacteria and the cell debris. The phagocytosis is basically one section of the endocytosis. Endocytosis can be of two types, phagocytosis or pinocytosis. Phagocytosis means if a solid material is engulfed, it is phagocytosis. Pinocytosis means that when a liquid material is engulfed, it is called pinocytosis. Pus is another term. Pus is basically the dead neutrophils and the liquid material. If you're having a face acne and all, if you squeeze it, sometimes a very whitish color thing can come out. It is basically pus. And if it is having a connective tissue capsule, it is called an abscess. Next, the immature form of neutrophils, they are unsegmented or less number of the nuclear lobes will be seen, and they are referred to as the juvenile cells or the band cell. Next, the pseudoneutrophils and heterophils are comparable to the neutrophils, and they are seen in rabbit as well as poultry. Pseudoneutrophils are seen in rabbit, where the heterophils are seen in poultry. This is a diagram depicting how these neutrophils will act. First, this is a normal situation where the neutrophils are going to the blood vessels or the WBCs are going to the blood vessels. And here you can see there is a broken skin surface and a pathogenic cell debris and some pathogens bacteria are here. This is an example, classical example of a skin wound you are having. After that, the neutrophils will closely align to the endothelial lining. This is the endothelium, which is the boundary of our blood vessel, and they will closely align. This is called the margination property. After that, they will come out through diapedesis. They will come out, the neutrophils will come out through the small openings, and once they are out, they will try to engulf the cell debris and the bacteria, which is called the phagocytosis. You can see here, these are the various forms. First, they are forming myeloblast, then promyeloblast, myelocyte, metamyelocyte, banded, immature neutrophils, and the mature neutrophils. One more thing, if there is a more amount of banded neutrophils are seen in the blood, it means that there is an active infection is going on. Okay. Next type of WBCs are the eosinophils. Eosinophils are called so because their granules will look red or reddish orange. That's why they are eosinophilic in nature and appear red in color. The eosinophils are increased in case of parasitism. The parasitic organisms are opsonized attacked by antibodies, and the eosinophils discharge their granular contents onto the surface of the opsonized parasite, thus causing lethal damage to the parasite, which means that some antibody is required as well as eosinophils are required in case of attacking a parasitic organism. These granules will contain enzyme. The enzymes of eosinophils are rich in antiheparin, histaminases, oxidases, peroxidases. Thus, they will act as anti-inflammatory and anti-allergic agent. This is a very important statement because eosinophils have antiheparin while the next cells, the basophils, are having heparin. Then next is eosinophilia, which is an increased number of eosinophils in circulation. The gastrointestinal parasitic infection, allergic disorders. You have heard, no? If some person is having an allergy, he might be having eosinophilia, as in bronchial asthma. In skin diseases like eczema and dermatitis, and eosinopenia is a decreased number of eosinophils in circulation. The condition occurs following stress, administration of ACTH or cortisol. ACTH is adenocorticotropic hormone. Next, the basophils. The basophils are having an irregularly shaped nuclei, and the granules will be stained blue by the basic dye. In the interstitium or the tissue, the basophils will enlarge and become mast cells. Basophils are seen in circulation, but when they go to the tissue, they will become very large and they become the tissue mast cells. Both the basophils and the mast cells will release heparin, which is an anticoagulant, and vasodilator substances like histamine, serotonin, and bradykinin. You know that when you are having an insect bites you, you will probably getting an itching sensation right there, and you will start itching, and there will be a localized basophilia or a mast cell will be there, and this will release the heparin and the vasodilator substances like histamine, serotonin, and bradykinin. This is the cause of an increased amount of irritation and hyperemia in that bite site. The vasodilators causes increased blood flow and hyperemia, which is the reason for the reddish color at the site of infection. And it is followed by an increased permeability of the plasma and proteins from the blood vessel. That's why always we will see a little bit of swelling there. The basophils and mast cells have receptors for immunoglobulin IgE associated with allergies. The basophilia, it is an increase in basophils and it is seen in allergies. Next, the monocytes. Monocytes are agranulocytes and they are the largest WBCs with a kidney or bean-shaped nucleus. They are also very motile, pinocytic, and also actively phagocytic. In the tissue, they become macrophages. Very important point. In the circulation, they are monocytes, but if they go to the tissue, they will become macrophages. They are they are very important during the chronic inflammatory reactions. The monocyte number increases in chronic infections because of this reason. Mechanism of action is that the macrophages, which are the tissue monocytes, kill the phagocytosed microbes by their acidic pH, bacteriostatic proteins, and degradative enzymes. They also produce hydrogen peroxide in greater quantity when comparison with the neutrophil. I have already said that neutrophils produce hydrogen peroxide, but monocytes or the macrophages produce more amount of this hydrogen peroxide in comparison to neutrophils. Next, very important WBCs are the lymphocytes. The lymphocytes can either be T lymphocytes or B lymphocytes based on the immune responses or based on their sizes. They can be large, which are immature, and small, which are mature. If after birth or the postnatal processing and differentiation of lymphoblasts are the precursors of lymphocytes, occur in the thymus gland. It is called T lymphocyte, capital T, T lymphocyte for thymus gland. If this postnatal processing and differentiation of lymphoblast occur in fetal liver, spleen, bursa of Fabricius in case of birds, and in the bone marrow, B lymphocytes are formed. Just think B for bursa and B for bone marrow. These two terms, postnatal processing and differentiation, you will study it in immunology sections in microbiology. But for the time being, you remember these 2 points. These lymphocytes then go into the circulation, and after their processing and differentiation in these respective places, this these cells will go to the circulation, and they will get localized in the spleen and the lymph node as T cells and B cells. In both the spleen and lymph nodes, you can see the T cells and B lymphocytes. Next, let's study a little bit more about T cells and B cells. T cells are of three categories: cytotoxic or the killer T cells, helper T cells, memory T cells. The cytotoxic T cells are the T cell receptors in these cases bind to the specific antigens, and the cytotoxic substances are released into the foreign material, example bacteria, viruses, and tissue cells. Here it is the cytotoxic T cells which are the cause for the organ rejection, and they are also very important in fighting the cancerous cells. Next, the helper T cells. The helper T cells are required for the activation of the cytotoxic T cells as well as the B cells. These cells, helper T cells, will produce some type of lymphokines. The lymphocyte produces this type of lymphocytes produces lymphokines. Example is the interleukin 2 or the IL-2. Next is the memory T cell. The memory T cell is basically, they are very long lived. They can live for years, and they will respond to the same antigen when exposed at a later date. For example, today if one antigen or one bacteria or one pathogenic organism enters your blood vessel, this these two cytotoxic T cells and helper T cells will act at that time, but memory T cells will remember it. These memory T cells will remember it, and they will remain dormant till and when another day or another month, after which you will get again the attack. Maybe after an year you will get this attack by this another one pathogenic organism. At that time, the memory T cells will remember this first attack, and they will produce they will respond to the same antigen when after a long period of time after the first attack. The T cells are involved in the cell mediated immunity. Very important point. Please remember it. Next, the B cells. After an exposure to the antigen, the B cell becomes activated. So they are called the activated B cells. The activated B cells can proliferate, and they can transform into either plasma cells, which are more in number, and very few memory B cells. There are two types of memory cells, either memory B cells or memory T cells. So the plasma cells are very important, that they are the one that produces the antibody. Antibodies are globulin molecules, also called the immunoglobulins, because of their important action in immunity, and they will inactivate the foreign substance because they are acting through antibodies. The B cell is very important in humoral type of immunity, where the T cells are important in cell mediated immunity. A same way in the T cells, T memory cells, here the B memory cells are also converted to the effector cells or by a later encounter with the same antigen. How does antibodies react, or the mechanism of action of antibodies? These bold points, please take it in, they are important. First, agglutination. Second, precipitation. 3, neutralization. 4, lysis. Agglutination. Agglutination means that antibody will bind to the multiple antigen. You see here it is agglutination. One antibody is binding to multiple. This antibody binding to 1, 2. This antibody binding to 1, 2. So one antibody will bind to multiple antigen, and then it will cause to the clumping together, thus preventing the spreading and easier for the macrophages to engulf them. For example, if there is just for one example, consider you are having so much of iron dust. Hmm? Iron dust is our, for example, I'm just making an example. If the iron dust is a bacteria and you are bringing a magnet and you are putting this magnet is the antibody, and if you put to the magnet, all this magnet will absorb the iron dust. Same way, here the antibody does this to the antigen is the bacteria or cell debris. The precipitation means antibody will attach to the soluble antigen, for example toxin or viruses, and they form large complexes that becomes insoluble. The neutralization is antibody will cover the toxic site on the toxin or viral particle, thus preventing them interacting with the host cell. Next, lysis. When antibodies bind to antigen on the surface of a cell or bacteria, they activate the complement system. This causes opsonization. Opsonization is nothing other than the foreign substances are made vulnerable to the phagocytosis by neutrophil and macrophages. That means when a cell surface is having, this is opsonization, when the bacteria, this is the bacteria and so many antibodies are covering it, and when the antibodies are covering it with the complement system will get activated, and this complement system, with the help of the complement system, this antibody attached bacteria will be engulfed by the neutrophil and the macrophages. And finally, when this is getting attached, this antibody is getting attached to the bacteria, there's a more potent chemotaxis of the neutrophil and macrophages to the injury site occurs, and it will causes more effective phagocytosis of neutrophils and macrophages. Next, very important discussion: hemostasis, or the prevention of the blood loss. Now we have discussed about RBCs, WBCs, the mechanism of action of both RBCs and WBCs in various aspects we have studied. The next important subtopic is the hemostasis, or the prevention of blood loss. Hemostasis refers to the body's natural process of stopping a bleeding or hemorrhage. It involves a complex series of events that occur in response to a blood vessel injury. Here I have depicted 5 steps. This is what is given in your Duke's Physiology textbook. First, when there's a vascular damage, there will be vascular constriction, and the platelets will get adhered to this small opening. Then more amounts of platelets will get recruited, and the platelet will form aggregations, and there will be a fibrin formation will be there, and there will be a primary hemostatic plug will be formed, and there is a, in the third figure there's a reinforcement of the plug formation with the fibrin, and there will be a thrombus will be formed, and here in the fourth figure there's a clot is getting retracted, which means that clot is becoming smaller, and activation of fibrinolysis, and in the finally there is an endothelial repair. The endothelium is basically, you know what it is, right? It is basically the cell cells which make up the blood vessel. The endothelium gets repaired, and total degradation of the fibrin clot. In the upcoming slides, we will see in detail how this is happening. For the action of this hemostasis, the most important cell or the cell fragment that are required are the platelets or the thrombocyte. We have discussed about RBCs and WBCs. RBCs are required for the oxygen transport. WBCs are required for acting against the upcoming or invading pathogenic organisms. What is the action of the platelet or the function of the platelet? Their function is nothing other than the prevention of blood loss. So platelets are formed from megakaryocyte. The megakaryocyte will get fragmented, and the fragments of the megakaryocytes are called the platelet or the thrombocyte. The mammalian platelets are non-nucleated, but in birds and reptiles they are nucleated. There will be a thick glycocalyx of the platelet cell membrane. You know, glycocalyx is the outer covering of the cell membrane. A cell is having a membrane, right? And it is having an outer glycocalyx. And it is having the platelet clotting factors like fibrinogen, factor 5, factor 7, and factor 9. You have probably, I think that you have heard about these factors. The fibrinogen is in inactive form, it is soluble in the plasma, while the fibrin is the active form and it is insoluble in plasma. The phospholipid of the cell membrane is the precursor for the prostaglandins and the platelet factors. Platelet cytoplasm has two types of granules. Please study this two types of granules, the alpha granule and dense granule. They have asked this question in this past ICR net exam. The alpha granules, which is having albumin, thromboglobulin, factor 5, fibronectin, platelet-derived growth factor, von Willebrand factor, factor 13, and the dense granules contains the ATP, ADP, serotonin, actin, myosin, calcium, mitochondria, and glycogen. You know, actin and myosin are also very important in another area of physiology. You might have heard it, the muscle physiology, right? So they are also very important in the platelets also. These are the normal platelet count in domestic animals. Platelets are mostly in lakhs, where in case of RBCs that will be million RBC. These are lakh RBC. Sorry, lakh platelets per microliter of blood. Where in case of WBCs that will be thousands per microliter of blood. Platelet reaction. So the important function of platelet is to prevent the blood loss. So how does the platelet will act in that situation? It is called the platelet reaction. The platelet will undergo structural changes at the site of injury, leading to the platelet reaction, and the granules will get released and creating a highly reactive surface for the thrombin and fibrin formation. The following subheadings which are marked in red are the platelet reaction steps. First, platelet adhesion. Once an endothelium is getting disrupted, a blood vessel is there, its endothelium is getting disrupted, the platelets will adhere, platelets will get sticked to this site, and this will form a monolayer. This triggers a shape change with the development of pseudopods. You know, pseudopods are false foot. So platelets will develop a false foot, and with the help of the false foot, each of the platelet will attach to each other and they will get adhered to the disrupted endothelium. And that adhesion happens. How does this adhesion happen? This is because the endothelium usually had a collagen that was concealed, that is not exposed. But when an injury is happening, the endothelium is getting disrupted. The collagen from the subendothelium, which means the endothelium will be there, and just below the endothelium there will be subendothelium, that when it is getting disrupted it will open up and there will be exposure of collagen and there will be exposure of fibronectin from the endothelial cells. These two things, the collagen and the fibronectin, causes the attraction of the platelets and causes the adhesion of the platelets. Further, the adhesion of the platelets is facilitated by the presence of the von Willebrand factor and the fibronectin in the platelet granules, which are released upon platelet activation. We will see what is platelet activation. Platelet activation is caused by various agonists like collagen, thrombin, ADP. They bind to the specific receptors activating intracellular messengers. The platelets also has got their messengers, sorry, receptors and messengers. So when a collagen, thrombin or ADP bind to these specific receptors on platelets, it will cause intracellular messenger production. The most important principal messenger is the thromboxane A2, TXA2. And the TXA2 is produced from the platelet membrane phospholipid. And the production of the TXA2 within the cell, within the platelet, causes an increased amount of calcium. Okay, and this causes the platelet activation. Okay, you have heard about aspirin, a drug. If you take aspirin, there are more chances of problems with the blood clotting. Blood will not clot. Why? This is because the aspirin blocks the TXA2, thromboxane A2 formation, and thus there will be a lesser amount of calcium production and calcium mobilization, and thus the platelet activation does not happen. Why calcium is required? Calcium is required for the next step, platelet release reaction. Because of the presence of the calcium, the granule, the platelet has got some granules, and within the granules so many components are there. I have mentioned in the previous slide. The granules will cluster in the platelet center due to the elevated calcium induced microtubular contraction. In the upcoming slide, I will depict a photo of it, a picture of it, and the extrusion of the granular content to the exterior through the open canalicular system. You see, this is an open canaliculae. This is the platelet release reaction, association with the adhesion and aggregation, fusion of the granule. See the granular membrane, the alpha granule, dense granule. The alpha granule is getting here in the photo, the picture, the alpha granule is attached to the open canalicular system membrane, and the granular content will be extruded. It goes out through the open canalicular system. The platelet aggregation is the last step of the platelet reaction. As the granular contents are out, high concentration of fibrinogen, which are needed to form the fibrin, fibronectin, von Willebrand factor, which are both fibronectin and von Willebrand factor, both are required for adhesion. Factor 5 and other proteins that assist the conversion of the prothrombin to thrombin at the platelet surface will be so high. So many components like fibrinogen, fibronectin, von Willebrand factor, factor 5, and many other proteins will come outside from this platelet to the from the inside. They will, all of these components will come outside. We already said that fibronectin and von Willebrand factor is required for more adhesion, and because of this, more and more amount of platelets will get adhered, sticky. They will become all sticky, and they will form the primary platelet plug. Very important word. The primary platelet plug is formed by the platelet reaction. Okay. Further, next is the actual blood coagulation or the blood clotting. You might have heard about this somewhat complicated form in which they are in textbooks. We can see the extrinsic and intrinsic mechanism. So the factors, before that, discussing that in detail, we have to know what is a procoagulant and an anticoagulant. A substance that promotes the coagulation is called a procoagulant, and substance that inhibit it is called an anticoagulant. Normally, the blood is having anticoagulant activity. If just imagine if the blood is having more amount of the coagulant, procoagulant activity, the blood will clot. So the blood is normally having an anticoagulant activity, and but if a blood vessel rupture, the procoagulant activity increases in the affected area, leading to the blood clotting. So I already said about a primary plug formed by the platelet adhesion, platelet reaction. The secondary hemostatic plug, it is otherwise called the clot. This question can ask in exam, ask be in exams. It is the stabilized platelet plug formed by the blood coagulation, which is stabilized by the formation of fibrin. This is a stabilized platelet plug. uh, and it is formed by the blood coagulation, which is stabilized by the formation of fibrin. So in the primary platelet for plaque, there is no fibrin, but in the secondary hemostatic plaque, there is a formation of the fibrin, which is insoluble in water. The cascade phenomenon. So the cascade phenomenon is most of the protein that participate in the hemostatic process circulate in the plasma as the inactive proenzyme. There are so many factors that are basically they are proteins. They are inactive form in the usual situation. The inactive proenzymes. They are inactive proenzymes, but they will undergo activation, and one when it is undergoing activation, it will activate the other one. It will activate the next one. Then it will activate the other one. Like it is a cascade. So this is the cascade phenomenon, and this will lead to a production of a larger response. Here you can see the intrinsic and extrinsic mechanism of the blood coagulation. The intrinsic mechanism or the endogenous mechanism or the contact activation pathway. Here the trigger is the contact with a foreign substance, not the intact vascular endothelium such as disrupted endothelium, a negatively charged surface or glass. Here the intrinsic mechanism is that here the blood is not the blood is not coming in contact with any tissue, but it is coming contact with some other substance like a disrupted endothelium or a negatively charged surface or glass. Here the process starts with a series of enzymatic reactions within the blood vessel to initiate coagulation, means no tissue is involved. Example, the blood which is drawn in a glass tube close to this mechanism. If you are taking a blood, taking some blood from after an injection, you will draw the blood, and if you take it to a glass tube, after some time you can see that the blood is getting clotted in that. This is because of the intrinsic or the endogenous or the contact activation pathway. But in the case of the extrinsic or the exogenous mechanism or the tissue factor pathway, the trigger is the contact of the blood from a damaged blood vessel with extravascular tissue factor. Very, very important point. The tissue thromboplastin, which is otherwise called the tissue factor. This is required for the extrinsic or exogenous mechanism. The tissue thromboplastin is typically absent in the bloodstream under the normal condition. Inside the blood, it is not seen, but it is released to the blood when the surrounding tissue is also damaged. Just imagine you are getting a knife cut on your finger. So you are having a tissue, and beneath the tissue there's a blood vessel. So both the tissue as well as the blood vessel is getting cut. So the tissue factor or the tissue thromboplastin is getting released to the blood vessel into the blood, and this will cause the starting of the blood clotting mechanism. Okay, so two pathways: the intrinsic or the endogenous mechanism, or the extrinsic or the exogenous mechanism, or the tissue factor pathway. If you compare between the intrinsic and the extrinsic pathway, the extrinsic pathway will be always more fast because there's an involvement of the tissue factor as well, and even a small amount of stimulus can produce a large amount of the final end product, the clot. The damaged tissue releases the tissue thromboplastin. This contains tissue thromboplastin is not a single thing. It is composed of phospholipids and glycoprotein, which acts as the proteolytic enzyme. But in the intrinsic mechanism, the injury to the blood actually affects the factor 12. This is also a very important point. In intrinsic pathway, it is the factor 12 that is getting activated, and when the factor 12 comes in contact with the collagen or the vegetable surface, it becomes a proteolytic enzyme called the activated factor 12. That's why it is called the contact activation pathway. Why? Because the factor 12 is already present in the blood, but when the tissue disruption, when the endothelium is disrupted, it is having it will expose the collagen. So when it is exposed to the collagen or a vegetable surface like a glass, then the factor 12 becomes activated. For this activation of the factor 12, there's a important substance called the precalciferin. Precalciferin is required for the activation of the factor 12 to its active form. These are the various clotting factors that I have depicted here. Uh, you might get questions. You please try to study important factors like factor 1 is fibrinogen, factor 2 is prothrombin, factor 3 is tissue thromboplastin, factor 4 is calcium, factor 5 the labile factor, factor 7 the stable factor, factor 8 the antihemophilic factor, factor 9 is the Christmas factor, factor 10 is the Stewart prober factor, factor 11 is the plasma thromboplastin antecedent, factor Factor 12 is the Hachiman factor. Factor 13 is the fibrin stabilizing factor. These are some other names of these same factors. Factor 3 is otherwise called the tissue factor. Factor 8 is otherwise called the antihemophilic factor A. Factor 9 is called the antihemophilic factor B. Factor 10 is called the Stewart factor. It is otherwise known as the Stewart-Prover factor. VWF or the von Willebrand factor, it is required precalciferin. It is called the Fletcher factor. HMWK, it is the high molecular weight kininogen, fibronectin. These are some of the important factors. And here you can see that I have added a tenase complex and the prothrombinase complex. This is also very important. You have to study which factors are in the tenase complex and which factor is the prothrombinase complex. This I will deal in the subsequent slides, but for the time being you study that factor 9 combined with the factor 8 both in their active forms with the phospholipids and calcium is the tenase complex. Factor 10 activated, factor 5 activated with the phospholipids and calcium forms the prothrombinase complex. This is the intrinsic pathway and the extrinsic pathway. Very easy. Intrinsic pathway and extrinsic pathway up to this stage they are separated, but after the activation of the factor 10, this is a common pathway of coagulation. Let's see what is happening. Very short form. For further details you can go through read the Duke's Physiology, either the 12th edition or the 13th edition. But for the time being, you can see that the intrinsic pathway, the factor 12 is getting activated. The activated factor 12 will further activate the factor 11. The activated factor 11 will activate factor 9. This happens in presence of calcium. Then the activated factor 9, factor 8A, factor, phospholipids and calcium. These four factors form what I have previously mentioned. All these factors forms the tenase complex. Factor 9A, factor 8A activated, phospholipids and calcium. This tenase complex is required for the activation of the factor 10 to factor 10A. Then the activated 10A, factor 5A, phospholipids and calcium. These four factors form the prothrombinase complex. The prothrombin is converted to thrombin by the prothrombinase complex. And the thrombin is another proteolytic enzyme and it converts the fibrinogen, the soluble, water soluble or the plasma soluble inactive fibrinogen is converted to the active fibrin monomer. And the fibrin monomers, this fibrin monomer will form the polymer, and this polymer is forming the fibrin clot, and the thrombin is again converting the factor 13 to its active form, factor 13A active, and this factor 13 active is required for the stabilized fibrin clot formation. Here you can see that in the extrinsic pathway, factor 7 is required. It starts with the factor 7. Factor 7 and the tissue factor or the tissue thromboplastin is required, and the factor 7 is converted to the active form, and this factor 7 A combined with the tissue factor with the calcium can act at 2 points. It can either cause the activation of the factor 9 to factor 9A active, or it can act on the factor 10, where it can activate factor 10 to the active for factor 10A active. Okay, so here the intrinsic pathway. Intrinsic pathway is initiated by the trauma inside the vascular system, such as blood damage to the blood vessel walls. Most important factors is the factor 12, 11, 9, 8, and other cofactors. While the extrinsic pathway, it is triggered by the external trauma that causes the blood to escape from the vascular system. Here, the tissue factor as well as the factor 7 are the most important factors. Now, you can see that following vascular damage, the tissue factor and the binding site for the factor 7, factor 9, and factor 10 are exposed on the surface of the endothelial cell in presence of calcium. The tissue factor and factor 7 complex forms active because they become the first active, and they can activate for the factor 9 and factor 10. The activated factor 9 is becoming a part of the 10x complex. So they will activate the factor 10 to its active form factor 10A. If this two factor 7 7A and the tissue factor, only if they are present, they produce a lesser response in compared to both of them combined. If both of them are present, for example, the tenase complex as well as the factor 7A and factor tissue factor, both of them are combined, it produces more amount of the prothrombin to thrombin production. Okay, that's written in this point. The rate of factor 10A formation by the 10S complex occurs at a faster rate than that produced by the tissue factor, factor 7A complex acting alone, providing an amplification in the thrombin generation. The thrombin is again acting on 2 points. Thrombin, apart from converting the fibrinogen to fibrin monomer, it can have a positive impact on the factor 8 and factor 5. Till these points, in these areas, there is nowhere I have written that how factor 8A is getting activated and how factor 5 is getting activated. It is by the production of thrombin, factor 8 gets activated and factor 5 gets activated. In the initial formation of thrombin accelerates factor 10A production by a positive feedback response that activates factor 8, a component of the tenase complex, and factor 5, a component of the prothrombinase complex. The next step of the clot, now the clot secondary plug is formed. The clot will retract. Clot retract means after the stabilization, the clot will shrink due to the platelet contractile protein. I have said that like in muscles, there's actin myosin is present, and apart from that, a thrombostenin is also present for the clot retraction. Why the clot is getting retracted? If the clot is getting retracted, we know that within the clot there is no blood flow as such. But if the clot becomes smaller, more area is available for the blood to flow, and blood contains nutrients. So the blood is required for the removal of the clot again, and this will allow the tissue to repair faster. So the proteins like thrombostenin, actin, and myosin are exposed when the platelet are activated. The activation causes the changes in the thrombostenin, actin, and myosin, and just like a muscle contraction, this will happen, and this will causes the clot to retract. As discussed, this process allows increased blood flow in the damaged area during tissue repair, and the clot retraction squeezes serum from the clot. You might have seen that sometimes if you itch on your clot, a clear fluid is coming out from the clot. It is nothing other than the serum. The next step is the clot growth. Clot retraction is opposite to the clot growth. Clot growth occurs once the blood coagulation starts. When the clot is starting to develop, it will initially it will become enlarged, right? A clot will start and it will become enlarged. and the clot retraction means the clot will shrink. So both of these are opposite things. Okay, initially there's a clot growth and finally there's a clot retraction. Clot growth causes the expansion of the clot into the surrounding blood. The clot growth ceases when the fast flowing blood removes the generated thrombin. Thrombin is required for the clot formation. Thrombin, when it is not absorbed by the fibrin and other activated factors, is also washed away by the blood. The effectiveness of the washed away thrombin is reduced due to dilution because it is getting diluted. When initially the thrombin was limited to a little bit area, clot will happen. But what if the thrombin is getting very easily washed away and it will get diluted in the blood? No more clot will happen. The natural anticoagulant substance in plasma is the antithrombin 3. Please remember this antithrombin 3. It prevents unwanted coagulation. These substances counteract the procoagulants in small quantities. So normally a little bit of the procoagulants will be seen in the blood. It has to be prevented by the presence of the anticoagulant like antithrombin 3 and thus maintains the balance in the blood coagulation. And at last, the fibrin, the clot that has been formed due to the fibrin has to be degraded. Okay, this is called the fibrinolysis, the degradation of the fibrin. Plasmin is required for this. Plasmin is a proteolytic enzyme. It is responsible for the fibrinolysis. Plasminogen is the inactive form of plasmin, which is seen in the plasma, and it gets trapped when the clot is formed. Within the clot, the plasminogen is present, and the plasminogen becomes activated to the plasmin form by the plasminogen activators seen in the blood as well as tissue. Tissue type plasminogen activator, that TPA, it is released by the endothelial cells, and the reason for this plasminogen tissue type plasminogen activator formation is the thrombin and the blood stasis. When a clot is formed, there's presence of thrombin and blood stasis, and because of thrombin and blood stasis, the tissue type plasminogen activator is formed, and the TPA. The TPA causes the breakdown. The plasmin, more amount of plasmin is formed. Then the TPA produces more amount of plasmin, and the plasmin causes the fibrin to degrade or break down into the fibrin degradation product, or the FDP. This diagram itself is self-explanatory, but the FDP is taken up by the mononuclear phagocytic cells, and they will remove the phagocytic they will phagocytose, and they will remove the cell debris as anywhere it can happen. And in the normal circulation, how the clotting is prevented? I already said that antithrombin 3 is a natural anticoagulant. Okay, so antithrombin 3 is present. Small amount of thrombin are normally present in the circulation. So antithrombin 3 blocks this action and inactivates the bound thrombin. Next is the endothelium smoothness. I have said that when the endothelium is broken, there will be exposure of collagen and fibronectin. So when the endothelium is smooth, it is maintained as such. There will be no more activation for the factor 12, which is very important in the intrinsic system. 3. A monomolecular layer of protein with a net negative charge which will repel the clotting factor and platelet. There will be a protein with a net negative charge on the endothelium. 4. Heparin, very important, which is the natural anticoagulant that is produced by the mast cells in the precapillary connective tissue. Mast cells are abundant in the lungs because lungs are having more chances of emboli formation, which is freely floating blood clots. So its effectiveness is in preventing the blood clots is by the action of it is combining with the antithrombin 3. The heparin will combine with antithrombin 3. So it becomes more active, more effective. The heparin antithrombin 3 complex removes the thrombin and factors 9, 10, 11, 13 from the circulation. And the fifth is the anticoagulant protein C pathway. Thrombin, due to its potential biological effects, is regulated to prevent excessive generation around damaged blood vessel. If thrombin is produced in large amount and if thrombin is going to blood in large amount, clot can happen anywhere. So we have to prevent it, right? So the anticoagulant protein C pathway is one such important thing. In case of endothelial cells and peripheral blood cells, there will be a presence of a membrane protein called thrombomodulin, TM. So when a vascular damage is happening, automatically thrombin is produced. But to prevent its excessive production, the thrombomodulin will bind to thrombin, or thrombin will bind to thrombomodulin. Once the thrombin is bound to the thrombomodulin, the thrombin will lose its activity to activate the platelets and clot to the fibrinogen. So that activity is lost, but it becomes an activator of the protein C. So when the activated protein C is formed, or the protein C gets activated, it will neutralize the activity of factor 5A and factor 8A. Okay, factor 5A and factor 8A, we know that are required in the prothrombinase and the tenase complex. So that is reduced. That activity is. Till now, we have studied about the naturally occurring anticoagulants, but you may have probably seen in hospitals or clinics that various colored cap tubes available. So this is very important. Every competitive exams there will be one question from this section, that what is the anticoagulant of choice for glucose estimation? What is the anticoagulant of choice for blood culture? What is the anticoagulant of choice for normal hematology, for the serological examination? So please study it. This will be asked in your exams. Little bit about them. Chelating agents, which means that they will bind to the calcium. So calcium is required for the coagulant mechanism, right? So when the calcium is not there, no more coagulation can happen. So chelating agents like trisodium citrate, sodium oxalate, ethylene diamine tetraacetic acid, either the sodium or the potassium EDTA, they will bind to the calcium and make them unavailable for the coagulation process. Second is the sodium fluoride and the potassium oxalate. Both are gray in color. Both tubes will be gray in color, and they can be used for the glucose estimation because it inhibits the enolase enzyme. Enolase enzyme causes the glucose utilization. I will assign one task to you people. Please check what is the other name of the enolase enzyme and what in which biochemical pathway the enolase enzyme is required. Anyway, the glucose levels will get stabilized. So sodium fluoride and potassium oxalate. Heparin mechanism I have explained already. Then how heparin is causing anticoagulant mechanism. But heparin is normally not used for the leukocyte enumeration because it will causes the cell clumping. The yellow is used for the acid citrate dextrose, and it is usually used for the DNA testing like DNA fingerprinting, paternal testing, etc. Next is a test for coagulation. Much is not given in Duke's physiology regarding this, but we have to know a little bit about the coagulation test. First is the clotting time, and next is the bleeding time. Clotting time is, or the coagulation time, is the duration blood takes to form the clot once it is withdrawn from the blood vessel. In most domestic animals, the clotting time is less than 5 minutes. Here, usually the capillary tube method is used. You may have done in practicals that you will take a capillary tube, you will suck or get some blood to blood will get absorbed to it, and after every 30 seconds or 1 minute you will break a little part of the capillary tube and see clot is formed or not. Next is the bleeding time. It is required, that is the time required for the bleeding to stop after an incision is made. Incision is made on the live skin as such. So the bleeding time is used to assess the primary hemostasis, the platelet plug formation, where the clotting time evaluates the secondary or the true clot formation. Important point, please remember it. Normally, the bleeding time is 2 to 5 minutes. Here, the bleeding time is usually made by the IV method, where the incision is made on the forearm with a blood pressure of 40 millimeter Hg is kept. Next is the partial thromboplastin time and activated partial thromboplastin time. Partial thromboplastin time is nowadays not much used, and the activated partial thromboplastin time is used. The partial thromboplastin time focuses on both the intrinsic and the common coagulation pathway. I have said that there are a intrinsic and extrinsic pathway, and both of them will combine together at one point, and there will be common coagulation pathway. So activated partial thromboplastin type, it focuses on the intrinsic and common coagulation pathway. It is more sensitive than the partial thrombo PTT, partial thromboplastin type. The only difference is that here we will use an extra activator. You see here the name is caveolin cephalin floating type, which is the traditional name for the APTT. APTT initially when it was found, it was called the caveolin cephalin because it is used as a activator. It is a negative charge containing factor, and this causes the activation of the factor 12. So we know that in the intrinsic pathway, factor 12 is involved, right? So to activate it, the caveolin or cephalin is used. So this is the APTT. Both APTT and PTT are measured in seconds. Next is the one-stage prothrombin type. In this test, plasma is activated using a mixture of tissue factor and phospholipids. Calcium is then added to initiate coagulation, and the time to clot formation is measured. So this is used to mostly check the extrinsic and the common pathway, because you know that in the extrinsic pathway, tissue factor or the tissue thromboplastin is required. So for the extrinsic and common pathway, one stage prothrombin time, which is measured in seconds, is used. Now let's learn a little bit about the coagulation defects or some problems in coagulation. First is a dicarbonyl poisoning. Dicarbonyl poisoning is a common thing in some countries where the sweet clover is fed, and the moldy sweet clover, there's a formation of a component called the dicarbonyl. The dicarbonyl causes the inefficient utilization of the vitamin K, leading to the inadequate formation of the prothrombin and factors 7, 9, and 10. It is also very closely related to the warfarin poisoning, which is a synthetic component used in rodent as a rodenticide, warfarin. Next is a thrombocytopenia. Thrombocytopenia can happen when there is a reduced amount of the platelet count. It can happen because of irradiation of the bone marrow, like people who are undergoing radiotherapy, uh, drug sensitivity, and some infections. And the very commonly heard diseases, the hemophilia. Hemophilia are hereditary hemorrhagic disease. Hemophilia A happens due to a deficiency of the factor 8. It is congenital, which means that it is present from birth. The species affected are dogs, but it has also been reported in cats and horses. Next disease is hemophilia B, which is caused by reduction in the factor 9. This disease is inherited, which means that it is passed down through generations. It is seen in dogs as well as it is seen in human beings. Next is the von Willebrand's disease. It resembles the hemophilia A, and it is characterized by reduction in the platelet function and factor 8 production. Nature, it is inherited. Factor 9 deficiency has been reported in cattle. It is also an inherited disease. Next, let's see a very interesting. This is an interesting diagram where you can see that Queen Victoria. You probably might know who is Queen Victoria. You can see that the if you look very closely, you can see that the men of this diagram, men depicted in this chart or diagram, are have died in a very young age. Why? This is because the Queen Victoria has been a carrier of the hemophilia B infection, not infections, I'm sorry, gene, and because of that, it is an X-linked gene. So defective factor 9 production, and all male princes, all of them are princes. They have died in very young ages, except for Prince Leopold. He has given he has been able to give birth to Princess Alice. So this is about hemophilia. And Next is the blood grouping. Blood grouping, you probably might know in human beings, we have heard A, B, B, O type of blood grouping. This is called ABO blood grouping system. Why they are called A, B, such things? This is because of the presence of some typical antigens on the cell surface. Okay, these antigens are either glycolipids or glycoproteins. It was Carl Landsteiner. He's the father of the blood groups. He in 1900 was the first to identify four blood groups, which are the A, B, AB, and O in human beings. You can see these are four different types of RBCs. If a patient is sorry, if a person is having A blood group, he is having antigen A present on it on the RBC. If he is having B blood group, antigen B is present. If he is AB, he is having antigen A and B. If he is O blood group, there's no antigen present. But if antigen A is present, the body will not produce antibody against antigen A. It will not produce antibody against antigen A, but it will produce antibody against antigen B. Why? Because already the body is having antigen A. Why should the body produce antibody against its own antigen? It will not happen. So the body will produce antibody against antigen B, which is absent in it. The same way, if the client is having or the human being is having B blood group, there will be production of anti-A antibody. If both of them are present, antigen A as well as antigen B are present, both are present, so no antibody will be produced. If nothing is present, no antigen is present, both antibodies will be produced. The antigens A and B are present on the surface of the RBCs and they are known as agglutinogens. And the plasma antibodies that cause agglutination are known as agglutinins. During the blood transfusion, if the blood between the recipient and donor are mismatched, there will be a reaction of antigen agglutinogen with the antibody agglutinin causing clumping or agglutination of the erythrocyte, finally leading to hemolysis of the RBC in the circulating blood. Hence, the AB blood group is having no antibody, no antibody. They are the universal recipient. Hence, the O blood group is having no antigens, no antigens. They are having no antigen. It is called the O blood group is called the universal donor. Okay, this you might have studied in genetics. The why this alleles of this blood group. If A blood group, the genotype will be AA or AO. If the B blood group, BB or BO. Sometimes you might have an IA, IAI, IO like that. So if it is O blood group, it will be uh, OO. Genotype will be OO. Next is the RH system of blood grouping. Why it is called RH? The RH system is called because of the rhesus monkey from which it was identified. This RH is the reason why your blood is called positive or negative. Okay, you might have seen that almost very normally many human beings are the majority of human beings are positive. So he or she might be A positive, B positive, or O positive. Very rare cases only human beings are negative. So the presence of this Rh antigen makes them positive or negative. These are the transmembrane proteins on red blood cells with the exposed loop. Basically, it means that there will be a cell surface will be there, right? There will be a cell surface, and this is the membrane, okay? And this is the transmembrane protein, which means that it enters the membrane, and it is having a loop, okay? This is what it is I was saying. Okay, so this was named after rhesus monkey. The RH system characteristics is that there are 6 RH antigens C, D, E, small c, small d, and E. But the D antigen is widely prevalent and it is more antigenic. So if you're having D antigen, it means that you are RH positive. Blood transfusion in case of RH system. Actually, there's no immediate reaction will not happen between the Rh positive and Rh negative individuals, but there will be a delayed antibody. The antibody, if you are an Rh negative person, you will be having antibody against this Rh antigens, right? So if you're getting a from an Rh positive person, you will get antibody will be developed very slowly after 2 to 4 weeks. And subsequent, if you are getting again the some blood transfusion again from an Rh positive individual, that means if you are an Rh negative person and if you are getting blood from an Rh positive person, in the first time it will not become a big problem. But if you are getting the blood again in the second time, already there are antibodies produced against this Rh antigen, there will be enhanced so much severe immune reaction will be seen. This is what called the delayed type of antibody reaction will be seen. So keeping this in mind, we can see that a condition called erythroblastosis fetalis, this is seen in human beings, and neonatal isoerythrolysis in horses. In the erythroblastosis fetalis, the maternal circulation, the mother is having Rh negative blood and the fetus is having Rh positive blood. The fetus gets this Rh positive blood from his or her father, and the fetal blood will enter the maternal circulation, and the mother will sense this. Mother's blood will sense this, and the uh, mother's body will produce antibodies against this Rh positive blood of the fetus, and because of this maternal anti-Rh antibody will cross the placenta and it will enter the fetus, and the fetus will get uh, cell lysis. The RBCs of the fetus will get lysis because of the agglutination, and this is what called erythroblastosis fetalis. Hmm? How we can prevent it? We can prevent it by passively immunizing the mother against the Rh positive factor. Uh, Means when the Rh positive factor is entering into the mother, we can immunize the mother against the antibody that is produced. Okay, the neonatal isoerythrolysis is the next important thing that is seen in horses. Horses have different type of blood systems A, C, D, K, P, Q. Then incompatibility can occur when the foal inherits a blood type from the sire that the mare recognizes as foreign, causing hemolysis, anemia, icterus, etc. The disease is caused by the destruction of the foal RBCs by the antibody complexes present in the colostrum of the dam. Then to treat this condition, we have to first identify the mare with the antibody and withhold the colostrum, because to the colostrum the antibody will pass to the fetus. Sorry, the foal means the young baby of a horse. The young one of a horse is called a foal. So to prevent it, we have to first identify which horse is having this, which mare or the mother is having this antibody rich colostrum, and we have to prevent that colostrum feeding in such situation. Now we are entering the almost the last portion of our discussion of blood, the plasma. The plasma is the non-cellular liquid part of the blood obtained when the coagulation is prevented during the blood drawing. What is the difference between the plasma and serum? Okay, in case of plasma, we will add an anticoagulant, for example, EDT or heparin, so that we will get the clotting factors, which are the proteins. The clotting factors are also seen in clotting factors are seen in plasma. So if the clotting factor is allowed to remove, if you remove the clotting factors from the plasma, it is called the serum. Okay, that means if we allow the blood to clot, and after that we will get a liquid up, that is the serum. So this is the difference between the plasma and the serum. So to collect serum, we will not add any anticoagulants. The component of the plasma, about 92 to 94% of the plasma is water. Then uh, it is also having, plasma is also having the protein. Then the usual percentage of protein is 6 to 8 percentage or 6 to 8 gram per deciliter. The plasma proteins I have given here in the table. Usually the plasma proteins are like in a range of 6 to 7. The in pink it is little bit higher, 7.9 to 8.9. In a ostrich, this is a table from your Muck's veterinary manual. So that is little bit lesser in compared to other species. And you can also see in the Duke's physiology, it is said that in humans, sheep, goats, and dogs, albumin predominates globally. But in cases of horse, pigs, cows, and cats, the relative proportion of albumin and globulin are nearly equal. The general ratio of albumin to globulin is 1.11 is to 0.7. So generally, albumin is higher in compared to globulin. Okay, the usual plasma proteins are albumin, globulin. Globulins are alpha 1, alpha 2, beta 1, beta 2, and gamma globulin, and fibrinogen. More amount of proteins are there, but these three are the important things. Next, plasma protein fractions. We have studied about antibodies, right? Basically, the gamma globulins are the immunoglobulins. They are the antibodies, okay? And they are produced by the lymphocytes and the plasma cells. And the lymphocyte and the plasma cells will produce it, and they will release it to the blood or the plasma. Or okay, so these are the five important immunoglobulins. I have given the important features of the more is there, but these are the very crucial important points. The rest you will study in the microbiology sections. That IgG is the predominant antibody in the blood and tissues. It crosses the placenta and it offers passive immunity to the fetus in case of rodents and primates. In large animal or the livestock, this doesn't happen. Antibodies will not penetrate to the placenta. IgM. That's why I have said previously that the colostrum has to be withheld in case of horse with that neonatal type of hemolysis in equines. IgM. IgM is the first antibody produced during the primary immune response. Important point. It causes the RBC agglutination reaction. IgA. It is the everywhere IgA is present. It is present in mucous membrane, saliva, tears, breast milk, but it will not cross the placental barrier in usual species except in livestock. IgG will cross the placental barrier. For example, in human being, which is a primate, IgG will cross the placental barrier, but IgA will not cross the placental barrier, but it will be seen in the colostrum or the breast milk. IgE, which is involved in the allergic reactions and parasitism. Earlier, I have said about it. It binds to the mast cells and basophils and triggers the release of the histamine. IgD, it is found on the surface of the B cells, functions in the activation of B cells. Next, the alpha and beta globulins serve as a substrate for the new substances and also perform transport functions. Example, lactoferrin, a globulin that transport iron. New substances means if a new protein has to be produced or new substance has to be produced, alpha and beta globulin are required. Plasma albumin, some of the globulins, and fibrinogen and other coagulation factors are formed in the liver. The balance of the globulins, including the gamma globulins, is formed in the lymph nodes and the mucosal tissue. Hence, the prothrombin type can be used as a liver function test. Why? Because prothrombin is a plasma protein, right? If prothrombin is in good amount, which means the liver is producing the plasma protein in a good amount. The prothrombin type is one of the coagulation tests I have said earlier. The albumin is very important in maintaining the colloidal osmotic pressure. It is otherwise called the oncotic pressure. The normal range of the oncotic pressure is 28 millimeter Hg. The molecular weight of fibrinogen, albumin, and globulin are 3 lakh Dalton, 70,000 Dalton, and 1 lakh 80,000 Dalton, respectively. Okay. And other plasma constituents are gases, oxygen, carbon dioxide, nitrogen, lipid, triglyceride, phospholipid, cholesterol, electrolyte, sodium, potassium, calcium, chloride, nutrients, glucose, amino acids, waste products, urea, creatinine, hormone, insulin, thyroid hormone, cortisol, enzymes, trace elements, and water. And this is the last slide. This was the first person. This was the person with which whom we started our discussion. He is James Harrison. He is called the man with the golden arm. His blood contains a rare and valuable antibody known as anti-D immunoglobulin. This anti-D antibody is used to create anti-D immunoglobulin, which is given to Rh negative pregnant women. I have said about the previously about immunizing the pregnant ladies who are having a fetus with the uh, Rh positive. So this treatment prevents Rh incompatibility issues between a mother and her unborn child, a condition that can lead to the hemolytic diseases of the newborn or the erythroblastosis fetalis. And he has donated blood more than 1,000 times. So if you are one such person who is afraid to donate blood or needle prick, you are afraid, uh, you please get motivated by him and start donating your blood once in a while. They will only collect 350 ml. 1 unit of blood is 350 ml. So try doing that and save more lives because blood. It is very important. Um, so that's it for today. You please contact me through mail in case of any suggestions. I know a little bit portions. This is a big topic. Uh, Without any intention, I might have left some area. So you can contact me if at all any clarifications are required. And that's it for today. Thank you. Have a good day.